In the last part of this lecture, we discussed about the anaerobic way of recycling NAD+. In this part now, we will be discussing about the aerobic cellular respiration, or the aerobic way to recycle the NAD+, the energy carriers, as well as what will happen to the pyruvate, that is the product of the glycolysis. So this aerobic cellular respiration allows a cell to completely oxidize glucose to water and carbon dioxide. So if you remember, from glycolysis, you have your glucose molecule, but at the end, you just got the 3-carbon pyruvate molecule. Now, since we usually say that the oxidation of glucose, in the oxidation of glucose, we have your glucose molecule as a reactant, and the product are water and carbon dioxide. So here, this part of this cellular respiration is where we will see how the water and carbon dioxide is formed. So in this part, we will be, or rather, the yield is 38 molecules of ATP per glucose. So this is a 19-fold increase in the amount of energy generated by the usual fermentation reaction from the last part. Because remember, in a fermentation reaction, the actual yield is just the yield from glycolysis, which is the 2 ATP. Net yield from glycolysis, 2 ATP per glucose molecule. In this aerobic cellular respiration, you have a much greater yield, which is 38 molecules of ATP. So in this aerobic respiration, you have one of the first step is pyruvate oxidation. So the pyruvate from the glycolysis, the pyruvate must be oxidized before it can enter this next part of this aerobic respiration, which is the citric acid cycle. So the enzyme pyruvate dehydrogenase is the one that's used to complete this task. So this is your pyruvate dehydrogenase enzyme. It's a protein, it's a large molecule. So this enzyme catalyzes the reaction where your pyruvate is basically processed or oxidized. So remember, pyruvate is 3 carbons. So 1 carbon is removed in the form of carbon dioxide. So you have here 2 carbon dioxide because you have 2 pyruvate molecules. So remember, you have your glucose coming from which is 6 carbons. At the very start of the glycolysis, you have 6 carbon glucose, and at the end of glycolysis, you have 2 molecules of pyruvate. So here is the 2 molecules of pyruvate, and a pyruvate has 3 carbons. So 1 carbon is removed in the form of carbon dioxide. So that's the first 2 carbon dioxides product of your respiration. And then you have another reduction of NAD+. So an additional reduction of NAD+ to NADH. Now the resulting reaction, or rather the resulting product, so when you remove the 2 carbons, you get, when you remove 1 carbon from pyruvate, you get the 2 carbon molecule called the acetate. So this acetate is added to your coenzyme A. Coenzyme is rather large, so it's not, this here is just a simplified representation, but it's actually a large molecule. So coenzyme A with the acetyl molecule, we call it the acetyl-CoA, acetyl coenzyme A. So this acetyl-CoA will be the one to enter the citric acid cycle. So here is the citric acid cycle, so also known as the Krebs cycle. So you have the release. So in this cycle, there is a release of energy from acetyl-CoA, and the released energy is transferred to electrons in carrier molecules as well. So aside from that, you release carbon dioxide and creates a little bit of ATP through substrate level phosphorylation. So this is called the cycle because at the end of each turn, you will regenerate oxaloacetate. And then each cycle will run twice for every glucose molecule because one acetyl-CoA enters the cycle at a time. So that means your cycle is running for 1 glucose molecule, which has 6 carbons, and it will end up with 2 acetyl-CoA molecules, and each acetyl-CoA will enter one at a time for the citric acid cycle. So what is this cycle? So it's a cyclic reaction. So at the first, you start with you enter the cycle through the acetyl-CoA. So the acetyl-CoA, or rather your coenzyme A, will deliver the acetyl molecules to your oxaloacetate. So this oxaloacetate will produce the citric acid. So that is why it's called the citric acid cycle, because the first molecule, the first resulting molecule here after the acetyl-CoA has entered is actually citric acid. So this citric acid will be converted to isocitrate, and then this is oxidized to oxalosuccinate. So you will generate here 1 NADH molecules. And then this oxalosuccinate will be 1 carbon dioxide from oxalosuccinate will be removed to form your alpha ketoglutarate. Then another oxidation. Another oxidation and another release of your carbon dioxide. So you will have here succinyl-CoA. And then succinyl-CoA will become succinate, then it becomes fumarate, malate, and then back to oxaloacetate. And then your reaction occurs again or begins again. So basically, it's a cyclic reaction. So you, at the end of this reaction, you will get how many NADH? So another reduction here of NAD+. So you have 1, 2, 3. So you have 3 NADH generated in this reaction, and then you also have 1 ATP molecules resulting from this reaction. This is from succinyl-CoA to succinate. So you will generate ATP here. So for one turn of the cycle, plus 2 carbon dioxide molecules. So by the end of this citric acid cycle, actually by the formation of succinyl-CoA, basically all your carbons from glucose have been generally used up. So how do you use it up? So basically 1 carbon dioxide, 1 pair of carbon dioxide from the oxidation of pyruvate to acetyl-CoA. So there's a release of carbon dioxide here, 2 carbon dioxide in particular, because you have 2 pyruvate molecules. And then this citric acid cycle, since it's running twice for every glucose, so you have here the release of carbon dioxide 1 times 2, so 2, and then another 2 here, 2 and 4. So basically you used up your 2, 4, and then the other 2, 1 from the acetyl-CoA to rather the formation of acetyl-CoA. So you have a total of 6. So by now you have produced your 6 carbon dioxide. So at the end of the citric acid cycle. However, our process doesn't end there, because again, here you have further produced more NADH, which are the electron carriers. So what happens to those electron carriers? So that will be on the next part of this cycle, which is the electron transport system. This is actually the oxidative phosphorylation stage. So the electron transport system, or the oxidative phosphorylation, is the last stage in the aerobic respiration. Actually, when I say oxidative phosphorylation, it's basically the mass production of ATP. And the electron transport system is different. They are not equal technically, but the process is basically through the electron transport system. The process of oxidative phosphorylation is through the electron transport system. So what is this system? So it's basically a set of transmembrane proteins that are basically that is, how should I say, responsible for generating a gradient. You will see later. So this process requires oxygen, and ETS is a series of membrane-bound proteins with increasing electron affinity. So you have here an activated carrier molecule from the citric acid cycle delivering the electrons. So what are these activated carrier molecules? These are the NADH. So this delivery of electrons, they first deliver the electrons to the first receiving protein in the electron transport chain. Some people call it electron transport chain because it's basically a chain. So you have a set of proteins passing electrons one after another. So this the passing when each protein passes an electron from the next protein to the next protein, there is a transport as well of protons, hydronium ions. You have a transport of protons, and this one generates the hydrogen ion gradient, which will be used to power the oxidative phosphorylation. It is also you will see here why oxygen is important. Oxygen is important here because oxygen is the terminal electron acceptor. So all of the electrons that the electron carrier, NADH, passes to the ETS system ends up with oxygen. And this oxygen, when oxygen receives this electron, it generates water. So here is your electron transport system. So here you start with NADH. So this is a more simplified version. So anyway, here you have NADH passing electrons to the first acceptor. This is the first protein in the chain. So in doing so, the protons are released from one side of the membrane to the other side of the membrane. And then it passes to the second protein called the ubiquinone. And the FADH2 is another electron carrier similar to NADH or NAD+. So another reduction or rather oxidation of FADH2. So passing another electron here. So the electron moves from protein complex 2 to protein complex 3. Another movement of proton from basically this one happens in the mitochondria. So the interior, this is in the mitochondria for animal cells and for the cells of how should I say, eukaryotic cells. But for the bacteria, that does oxidative phosphorylation, they do it in their own cell membrane. So anyway, so here you have another proton movement. Whenever the electron is passed from one protein to another, there's a movement of electron outside, to the outside of the membrane, to the outer side of the membrane. And then you have another protein from complex 3 to cytochrome C. And then you have here your oxygen. So this is the last complex, protein complex 4. So this protein complex 4 will then pass the electron to oxygen. So oxygen is the final electron acceptor. The oxygen here is basically the radical form of the oxygen. And it will join with the other hydrogen ions to form your water molecule. So here is the water molecule from the net reaction of glycolysis, or rather glucose oxidation, which is the oxygen, one oxygen for every one electron that is being passed. So you basically have 6 oxygens to form the 6 water molecules. So basically that's how your electron carriers are regenerated. So the regeneration occurs here. Every NADH will pass on the electron they have from the glycolysis reaction and even from the citric acid cycle. It will pass their they will pass the electrons to the protein complexes in the electron transport chain. And in doing so, they will become oxidized back to NAD+. This NAD+ can be reused for in another set of glycolysis. So anyway, so what happens then? How about the ATP? Where does the ATP come from? The ATP from oxidative phosphorylation, this is oxidative phosphorylation, comes from this gradient. So every time that the electron is passed from one protein complex to another, there is a movement of protons. Up until you now generate a gradient, there is a greater concentration of protons on one side and there is a less concentration on the other side. So this is there is what you call a pressure, a chemiosmotic pressure now happening here, that the excess of the hydrogen ions on one side of the membranes wants to go back to the other side of the membrane. So basically the cell utilizes that, so they have this ATP synthase. So here is the ATP synthase, which is basically the way that protons, it actually rotates. It's actually a rotating motor. So the hydrogen ion, it's like a windmill or even a water mill. So the proton, they enter through the rotation of this ATP synthase, and in doing so, every rotation, there is a yield as the synthase allows the passage of the protons along the concentration gradient inside the cell. So every rotation, you will produce ATP. So there's a mass, basically it's a mass production of ATP. So there's a lot of ATP being produced here. So the exact molecule per molecule count is quite difficult. It's actually not consistent in every experimental procedure, but the general output is around 36 to 38 ATPs for every glucose molecule being utilized in this electron system, because unlike unlike substrate level phosphorylation, where you just have a direct transfer of phosphates from a high energy molecule like phosphoenol pyruvate to ADP to form your ATP, so that's basically 1:1. Here is a more indirect formation of ATP, because you will first generate an electron gradient, and that gradient must first be established, and that gradient will then power up this machine to mass produce your ATP. So basically, that's your oxidative phosphorylation through the electron transport chain or the electron transport system. So basically, that's our oxidative phosphorylation. So accounting that, so if you account the ATPs here, so from glycolysis, you have a net production of 2 ATPs, a net production of 2 NADH molecules. And then during pyruvate oxidation, just 2 NADH here. In the citric acid cycle, you generate 2 ATPs. That is 1 ATP for every run of the cycle. And then you have 6 NADH. That's 3 NADH for every run. And then FADH2 in the citric acid cycle as well. So 2. That is 1 FADH2 for every run. So the accounting, how do they know it's 38? So from ATP, direct ATP production, you have a total of 4 ATP. And then the NADH that comes from your these processes, it generates about around 3 ATPs per NADH. So you have a total of 30 ATPs. And then for FADH2, you have around 2 ATP in one. This is from one source. This is from our textbook. So 2 FADH2, rather 2 ATP for every, or rather 2 ATP for every FADH2. So you have 2 times, so that's 4 ATP. So counting them off, you have a net total of 38. So if you look at a different textbook, some of them counts that NADH produces 2.5. FADH2 is just 1.5 or even 1. So it depends on the textbook, but the general calculation is around 36 to 38 ATP. So why is it different? Again, it's difficult to account the direct amount because it depends on the experimental calculation or experimental procedure done to actually measure them, as well as of course the different organisms in study. So you have and another things, because it's very indirect, the formation of the gradient is an indirect way to produce the ATP. So it's if you look at different textbook, it might be different. So that is our cellular respiration. So overall, starting from glycolysis, you have basically 3 steps. This is for aerobic cellular respiration. So starting with glycolysis, you produced pyruvate here, and then you end up with the citric acid cycle, and finally the electron transport chain. So this is the aerobic cellular respiration. If you look at the anaerobic cellular respiration, which is fermentation, it shares the same starting point, which is this glycolysis, but instead of going to the citric acid cycle, it goes to the fermentation pathway where you produce either ethanol or lactic acid. So basically, this is our the general overview of the cellular respiration. So in the next part of the lecture, we will look at a different catabolism of other biomolecules.